Part 2 of the senses begins with the ear and hearing. So what we call the ear is the visible portion of the ear. It's actually called the auricle or pinna, but the purpose of that ear is to collect and direct the sound waves toward the middle ear. The middle ear is an air-filled cavity. That ear has elastic cartilage and it gives it some flexibility. And there's that word auricle. That is what your ear is. That canal into your eardrum is called the external acoustic meatus, and it goes right through the temporal zone. I'm sure you recall that term meatus. It's lined with hair and ceruminous glands, and ceruminous glands secrete cerumen, which is earwax. Cerumen's got a purpose. It slows microorganism growth, reduces the chance for infection, and it prevents objects and insects from getting into the ear. So here is a great picture of the ear. You can see the auricle is the outer ear, and then the external acoustic meatus is also part of the outer or external ear, and that ends at the tympanic membrane. The tympanic membrane is the eardrum. If you've ever looked at a frog, frogs have round, smooth spots on the side of their head, and that is their tympanic membrane right on their skin. So just past the tympanic membrane is the middle ear or tympanic cavity filled with air. That tympanic membrane is a thin, semi-transparent sheet. And this middle ear is connected to the pharynx. The pharynx is the back of the throat. It's connected to the pharynx by an auditory tube or eustachian tube. The purpose of this tube is to equalize the pressure on both sides of the tympanic membrane. If there's more pressure on one side of the membrane than the other side, such as when you go into the water and you feel that pressure on your ears, there's more water pressure than there is air pressure inside, and that does not allow the tympanic membrane to beat or to move as it should in order to hear sound correctly. So we have to equalize the air pressure on both sides of that membrane. The problem with this auditory tube is it can allow bacteria to travel from the back of the throat to the middle ear, causing ear infections called otitis media. This middle ear contains what are called auditory ossicles. An ossicle is a bone, and these auditory ossicles are the smallest bones in the body. You knew them growing up possibly as hammer, anvil, and stirrup, but now you'll learn that they are called the malleus, incus, and stapes in that order. So starting from the tympanic membrane is the malleus, then comes the incus, which attaches to the malleus, and then the stapes. These tiny little bones are in the middle ear. They have joints. Those articulations between the bones are the smallest synovial joints in the body. And we'll see that there's skeletal muscles on the malleus and stapes to prevent too much vibration of that tympanic membrane. So here you can see, here's the tympanic membrane, the eardrum. Attached to the tympanic membrane is the malleus, then the incus, and then the stapes, and here's a tiny muscle. There are also stabilizing ligaments in here, and this is an air-filled cavity. This is the auditory tube going to the back of the throat. The internal ear contains the sensory organs for hearing and equilibrium. It receives sound that becomes amplified from the middle ear, and what we see when we look at that inner ear is a snail-shaped object. It's bony, and it is called the Boney Labyrinth. So this Boney Labyrinth, let's see, there's a picture of it. The tan color is the bone, and they're allowing you to see through that bone to what's inside, which is the membranous labyrinth. So this is the snail-shaped bony labyrinth. and here is what are called semicircular canals. So the Boney Labyrinth is a shell of dense bone. It surrounds the membranous labyrinth, and it's composed of 3 parts to learn: the semicircular canals, the vestibule, and the cochlea. The cochlea is for hearing. There will be a lot of terminology in here, some of which we can kind of ignore. For example, there are 2 different kinds of fluids in the ear called perilymph and endolymph, and we don't distinguish between them. We don't even talk about them very much. So here are the 3 parts for you to know. The Boney Labyrinth, of the Boney Labyrinth is the semicircular canals. So here are these 3 circular canals, and they are, like X, Y, and Z axis in mathematics. And then this region here, kind of a lump, big lump here. I mean, we're looking through the bone, but it would look like a big bony lump here, is the vestibule. And then this snail-shaped part is the cochlea. You may have heard of cochlear implants. That is for hearing. So this cochlea here is the organ. Inside the cochlea is the organ of hearing. And these 2 regions here, the semicircular canals and the vestibule, are for different kinds of equilibrium. So if you take away that Boney Labyrinth, you're going to have this membranous labyrinth underneath. Again, the cochlea here is for hearing. Now inside, this is membranous, so it's the cochlear duct. Over here on the other side is the semicircular ducts, and these are within the semicircular canals, and these monitor rotation in 3 different planes, like the mathematics graph, X, Y, and Z, those coordinates. And then the vestibule here contains the utricle and saccule, and we won't talk too much about those, but the receptors here are sensitive to gravity and linear acceleration. So the semicircular canals are for rotational movement. The vestibule here is for gravity and linear acceleration, and the cochlea is for hearing. We're going to skip some of this. The semicircular canal ducts contain hair cells, so we see hair cells again. We don't need to know the names of these cells. Each duct has an ampulla. Again, we're not going to get too detailed on this. I'm going to explain it to you, but the testing won't be too detailed on this. Each duct has an ampulla, and receptors in the specific region of the ampulla are called the crista ampullaris. And so in here in the crista ampullaris is a region that contains hair cells. I'll show you that. So here's the crista ampullaris here. Here's the cupula, and here are the hair cells, and they're kind of embedded in this gel here. So we're looking at this ampulla here. There's an ampulla associated with each one of these semicircular canals. And this is for rotational motion. So you'll see how if you turn your head one way, the fluid will move in a certain direction, stimulating those hair cells. Rotation of the head causes movement of the endolymph, the fluid that's inside, moving the cupula to the side and distorting those hair cells. So movement in one direction will stimulate hair cells, and movement in an opposite direction will inhibit hair cells, and no movement at all will return the hair cells to normal. So in your ampulla, this is your cupula, is your hair cells, and movement is going to cause stimulation of those hair cells. So each one of those semicircular canals responds to one rotational motion, such as shaking your head no, nodding your head yes, and then tilting your head side to side. Those are the motions that are detected by the semicircular canals. Now, in the utricle and saccule, it's a little bit different. In there are also hair cells, but there are also tiny little stones called ear stones. They're made of calcium carbonate. They're crystals. And these crystals are in this gel, and when the crystals move during motion, they will stimulate the hair cells. So here are the crystals. Here, they're called otoliths. Lith means stone, oto is ear, and then here are the hair cells. So when your head is upright, the otoliths are on top of the membrane, and the weight presses on the surface and it pushes the hair cells down. But if you tilt your head back, gravity is going to move those tiny little bones downhill, and it's going to distort those hair cells, and you get the perception of linear acceleration, for example. So that's all we're going to say about equilibrium. Basically, there's rotational equilibrium with the semicircular canals and linear acceleration and gravity in the vestibule. Now we want to talk about the inner ear, the cochlea organ of hearing. And so this is a picture of that cochlea kind of uncurled. Remember, it's a snail type shape. So here we've uncoiled it just a little bit. So this, the cochlear duct is a long coiled tube. So inside here, see the cochlear duct. This whole region here is the cochlear duct, and it's surrounded by two chambers, and they're filled with fluid. Now this upper chamber is called the scala vestibuli, and the lower chamber is called the scala tympani. You are not expected to remember these details. I hope you can understand how we hear and just get the gist of it. Okay, again, there's hair cells related to hearing, and the hair cells are going to be in this cochlear duct. And the main organ of hearing is called the spiral organ, or the organ of Corti. Now, this organ of Corti runs the length of the cochlear duct. So let's look here. In this picture, the organ of Corti is pink. And you can see the hair cells coming out. This is a picture from our textbook. I think it's a little bit confusing. If you get it, fine. It does show good view of the cochlear nerve, but I believe this previous picture is a little bit better in terms of understanding hearing. And here is a sectional view of the cochlear spiral, an actual photograph. So let's get back to our original picture. So when sound comes in the ear, and we'll talk more about this in a little bit, it's going to strike the tympanic membrane and cause the tympanic membrane to vibrate. The malleus is connected to the tympanic membrane, so these bones also vibrate. This last bone, the stapes, is on a membranous circle here called the oval window, and it will knock into that oval window. Well, there's fluid in here, so if you hit the oval window, you're going to cause the fluid to move. Now, this was the scala vestibuli. Okay, this was the scala vestibuli. There's a membrane here called the vestibular membrane. And then we have the organ of Corti, this pink one. Then we have the basilar membrane, and then the scala tympani. The spiral organ located in the cochlear duct is on the basilar membrane. So let's look for that basilar membrane right here. Okay. The spiral organ's right on that membrane. And the processes of the spiral organ, those hair cells, are in contact with an overlying tectorial membrane. So I know there's just so much going on here, but bear with me, bear with me. So you've got this tectorial membrane and the hair cells. Sound waves at the tympanic membrane trigger pressure changes in the perilymph. The basilar membrane bounces up and down in response to pressure changes. The hair cell processes push against the tectorial membrane, and this stimulates a nerve impulse. So let's go back. We'll go back to here. So what happens here is that the vibrations strike the scala vestibuli and cause movement of the fluid in here. There's pressure waves in here. Depending on the frequency of the sound, it crosses over to the scala tympani. The pressure wave kind of crosses over. And where it crosses over, the organ of Corti hair cells are distorted. Then that pressure wave kind of comes out and escapes down here from the round window. So you strike the oval window, cause a pressure wave to move along here and cross over, and where it crosses over, that organ of Corti, the hair cells stimulate the tectorial membrane and stimulate a nerve impulse. And then that wave is dissipated out the other end. So let's look a little bit at sound. Sound is waves of pressure conducted through the air. In each wave, the area of compressed molecules and area of molecules. In each wave, and there's an area of compressed molecules, so you can see the blue compressed molecules, and an area of molecules farther apart. The distance between these is called the wavelength. So here the sound is entering the ear. Well, the frequency of the wave is the number passing a fixed point in a given time. So the frequency of sound is measured in cycles per second, or hertz. You've probably heard of hertz. Hertz. The pitch of the sound is really the frequency. Sound waves all travel at the same speed. So higher frequency means shorter wavelengths. A high pitch is, for example, high frequency sound would be 15,000 hertz, and a low frequency sound would be 100 hertz or less. So here's our wavelength. If you have more of these waves per second, you've got a higher frequency. Now the amplitude is the amount of energy, that is the intensity or how loud it is, and that's measured in decibels, and that's something you should be pretty aware of. A quiet library of soft whisper is about 30 decibels. There's nothing dangerous to our ears about that. But if you're in busy traffic or a noisy restaurant, if you were there for 8 hours, you'd probably have some damage. Subway, I've heard some incredibly loud subways. I think an hour in that subway would hurt your ears. So you can see how susceptible our ears are to damage. And eventually you can lose hearing. It's cumulative. So I get so angry when I see parents taking their children to very loud concerts without protecting their ears, because they are damaging their children's hearing for the future. So when we look at our ears, the vibrations come in at the oval window, and if it crosses over here, it's high frequency. If it keeps going further, it's lower frequency. Now, when people lose their hearing, often they lose the high frequency sounds. So women are harder to hear. If you want someone who's hard of hearing to hear you, you could talk in a lower sound, a lower pitch. the events involved in hearing. So let's go over all these again. The sound waves arrive at the tympanic membrane. The tympanic membrane vibrates, causing the auditory ossicles to vibrate. The movement of the stapes hits the oval window and causes a pressure wave in the scala vestibuli. The pressure wave distorts the basilar membrane on the way to the round window. Vibration of the basilar membrane causes vibration of the hair cells against the tectorial membrane. And then this information is gathered by the cochlear branch of cranial nerve number 8.